through a bit more that way, and then there's more time for you guys to ask questions and for discussions to happen. Oh, they were my slides. Okay. So what we're going to go through today is an analysis of two different chip targets, and we're going to use contrasting approaches for both of them. I spent a couple of years working in a breast cancer laboratory, or a breast cancer research group, and they were particularly interested in the activity of the androgen receptor when subject to testosterone and how it moves the estrogen receptor away in breast cancer. Because their big paper, which is linked there, showed that the androgen receptor will move the estrogen receptor away from the proliferative sites and will slow down the out of control proliferation in breast cancer. And so they were obviously searching for um, some SARMs, selective androgen receptor modulators, as a therapeutic treatment for breast cancers. So that was the context. They published that paper like a month before I joined the group, so I didn't work on that paper, but I have seen all of the data for that paper, and um, it was part of what motivated me to write the package because I could see what they did, and I could see that they didn't. The lab tech did it, and she was just one of those supremely gifted people, just incredibly capable, but she wasn't a bioinformatician, and so there were just some nuances that were missed, particularly when it comes to normalization. So we've got two chip targets, the estrogen receptor and then the histone mark, H3K27AC, which is a marker of active transcription or an enhancer. These samples are from the same passages and same experiments, so they treated the same sample with testosterone or dihydrotestosterone, and then they separated it and did a chip pulling the ER out and then a chip pulling the H3K out after splitting the sample. So they're actually kind of matched samples here. Who has done any ChIP-Seq before? Oh, awesome. You guys are going to ask me all good questions. So the rest, no ChIP-Seq before at all. Who's here hoping to try it with a TAC-Seq? Um, yeah, I thought there might be one or two of you. I've never done a TAC-Seq before, but a lot of people have talked to me about using this for the TAC-Seq, and I think the counting with a TAC just might take a bit more care because you've got the TN5 things. Like I've never worked on a TAC-Seq. But then I was talking to someone who works in attack at BioC Asia last year, and they went, it doesn't really matter. It's just counting, looking at differences in counting. It's not going to matter. So when it comes to attack, I have never tried it with attack, but it should work. Anyway, back to the script. So a ChIP-Seq primer, just for those who are not familiar with ChIP-Seq as the basic concept, we've got all our transcription factors or histones bound to the DNA. We perform cross-linking, and then you've got immunoprecipitation, which is the IP in the chip, and the CH is the chromatin. And then we pull that down, and we sequence the little fragments that are antibodies pulled down, and we assume that those bits of DNA were bound by our protein of interest. Big assumption, but it generally works. In most data sets, and one of the data sets today we'll particularly look at this, there's often an input sample where you just pull down IgG without the specificity of your target to get an idea of background noise. And so we'll come across that, well, we'll see it briefly in the H3K27. So we're all basically happy with the broad concept of what ChIP-Seq actually is. I had a great discussion with Damien over lunch, and he was saying, why are you doing ChIP-Seq? There's all these new technologies, cut and run, cut and tag, and they're much better. My perspective on that was absolutely yes. When you're calling peaks, yes. But when you're doing differential signal analysis, which is what we'll be doing today, where we're comparing across samples, what we're going to be doing is we're going to be having a fixed width peak. So we'll define the peak center and then spread out from that peak center. And so if you've used cut and run and got a really nice tight peak, then maybe you want to bring in your range or something But in reality, the peaks are going to be nicer, but when we compare across samples, I don't think it's going to make too much difference. Has anyone used cut and tag, cut and run, any of those techniques? Okay, cool. I haven't. Our lab tech in this group was just amazing at ChIP-Seq and they didn't want to change because they figured they knew how to do one thing. And so they were staying with that. Okay, in terms of what I'm hoping you guys all know today, um, we're all familiar with the genomic ranges object. I might ask if anyone's never seen a genomic ranges object before. Okay, you'll see them. And they're really cool. Like they're one of my favorite things in bioconductor space. They're just beautifully efficient and they're lovely to work with. And then there's a genomic ranges list, which is obviously a list of genomic ranges objects, but they do some special things as well. What about summarized experiments? Anyone not seen a summarized experiment? Yes. Okay. So we'll actually be looking at a ranged summarized experiment which the ranges are associated with each of the regions. ggplot2. Anyone here not used ggplot2 before? Awesome. And are we all familiar with the pipe? We're familiar with the differences between base pipe and the Magritte pipe. I've gone with the Magritte pipe for consistency all the way through this workshop because there are moments where you can use the dot to represent the object you've passed, and I really wanted to do that, just like twice. And so I thought I'm going to use the same pipe for the entire workshop just because of those two little moments. But the package will work with either pipe. And that's really what gets me to why I developed the package extraChIPs. So I was originally putting together a big snakemake workflow. I called it Gravy because it was gene regulatory analysis with variable inputs. That was my joke name. And of course, if you're an Australian, you'll understand that extraChIPs with gravy is hilarious. But in America, they call them fries, and it's not as funny over there, but we call them chips. Um, so I put together this snakemake workflow, and as I was building it, I found all these functions that I wanted, and I put them into a package, and that's where it evolved from. I never published that workflow. I made the mistake of having a workflow that did too much, and it overwhelmed the people I was working with at the time. It was basically you could do an entire PhD project overnight. And that was too much information. So it never got published. But that was what I was doing. Uh, my focus in the package was on differential signal analysis, particularly in more than one comparison. So the group I was working with was often looking at the androgen receptor and the estrogen receptor and histone marks. Plus, there was RNA-seq data associated with it as well. And the workflow was about integrating everything like that. But we're just going to look at the differential signal analysis. Do you understand what I mean by differential signal analysis? That's what I call it. Some people call it differential binding analysis, but I think with, histone marks and stuff, binding was a bit funny. There are a few functions that are happening under the hood for what's happening today, and I wanted to point them out because I like them as functions, but we probably won't interact with them a lot. If you're familiar with genomic ranges objects, you might have come across the reduce, set diff, and union functions. I've put an MC at the end of them, and they will retain the MCols when you do the operations. If you've ever done those operations before and you lose the MCols and you go, and then you've got to map it back, I've just done that mapping back. So I find them really quite useful. I'm not particularly working in the ChIP-Seq space at the moment, but I do come back and use those functions quite regularly. I wrote this before the tidy omics people really kind of started their voyage to great success. So I wrote a quick little parser to parse a GRanges object into a tibble. What it does is it puts the genomic ranges part just in a single character vector column called range. And then all the MCols are retained. I found that just a really simple, quick hack to do my plotting, which is what I wanted to do, and make my summary tables. You can turn that off and just have it look like a normal tibble where you've got the, seq levels, start, end, width, and all that just by set ranges car equals fault, and it'll work. I've got some plotting assay functions, which we will see. And then best overlap and prop overlap are for when you've got a range and you want to compare it to a feature. Like what feature does this range best overlap, or what proportion of it overlaps a feature. The other two functions. So we'll see the last two a little bit, but the first two we'll probably skip over in the workflow. Okay, so let's actually start our journey. Now we can all just kick this off with your the resource that you have already installed. So if you just do a Control Shift Enter or Control Alt C, whatever operating system you're on, load the packages in your workspace. I'm keen for you guys to execute all of these lines of code. I've put them all together. So I've executed them when I wrote this presentation and when I wrote the workshop, so they should all work. Just one point to note is if you're using this after the workshop, change extraChIPs workshop just to extraChIPs, because you won't need the extra data that I've incorporated. Okay, so all of our packages have loaded. We're happy. Yeah, I'm going to keep asking you questions all afternoon. Sorry. I'm just one of those people. Everything worked? Oh, excellent, excellent, excellent. I believe if you're on the compute resources that are provided, in the vignettes folder, it will be called something. Yeah, that will be it. Yeah, extraChIPs workshop.rmd, that's the one. So all the packages will be installed. That's one of the advantages of using this system. So I'm hoping that when we all execute this line of code, this chunk of code, that everything works and we'll at least be off to a good start, without too many technical dramas. My little favorite trick at the end there is I like the black and white theme with ggplot. I can't stand the gray background, so I put that. I forget it's there most of the time. But
issues and Stevie's talking, feel free to gesture over to me as well, and I'll come round and help.
Yeah, yeah. Okay, so although I'd love to say, all right, let's do the ChIP-Seq analysis. Here's the peaks, here's everything. I like to take a moment just to get everything right and make sure that our analysis is going to proceed smoothly. And so all of the data that we have here was aligned to HG19 or GSEH37. And so we're going to use that as our reference. And I've got a little hack function there called define seq info. It's part of extraChIPs. Just tell it what the genome is. I've got all the lengths lurking behind the function. If you want scaffolds, don't use that function. But if you're just going, I just want a quick seq info object that has all the right lengths and just the main chromosomes, go for it. I think I've left the mitochondrial off, but you can include it just by adding that to an argument there. And I've put CHR equals true, so you get the CHR prefix. So it's a quick and easy way to get a seq info object, quick and dirty way. And you can manipulate it however you want. It's probably safest to call a seq info object from a BAM file when you're working with the data, but this works pretty well for this actual workflow and the real world actually too. So are you guys familiar with seq info objects? Is that a strange looking structure? I know some people are going to be really familiar with seq info objects. All it does is defines the genome that we're working with. So you can see we've got chromosome 1, it's this long, it's not circular, and this is the genome name it came from. A lot of the genomic ranges and summarized experiments objects have these as a foundational structure, and getting that right saves you a whole lot of trouble downstream. And it's for those who've worked with genomic ranges before, and sometimes things just take a moment to sort out. So that's how I would start my analysis off, just by defining a seq info object. And there's a trick that you'll see in a minute, which is why I do it that way as well. We also want to load in genes and transcripts and exons, those annotations. Now, I've given you a scaled back version of the GTF. It is the entire set of annotations on the main chromosomes, but I've left out a whole lot of things that I didn't want. That way it would fit on your machine. So we'll load that in using import GFF from our track layer. And then we're going to put that GTF, the seq info object underlying that. That means when we're comparing ranges between two objects, they'll have the same genome as the core foundation, and they won't spit out any weird errors saying these genomes don't match. And that's why we get these seq info objects clean and clear at the start. So that's all worked for everybody. Yep, great. An enthusiastic thumbs up. I appreciate that. Thank you. All right. Um, what I'm going to do next is I'm going to split that GTF by the feature type. So I want to have a genomic ranges list at this point in time. One's going to be exons, another one's going to be genes, another one's going to be transcripts. That just helps working through the package. It's still the same data, but it's just split nicely. And then I'm going to use that to define a whole lot of genomic regions here. There's a little function I've put in called define regions. And if we give it all of the genomic coordinates for the genes, all of the genomic coordinates for our transcripts, and all of the genomic coordinates for our exons, it will spit out um, the entire genome allocated as a promoter, an upstream promoter, a gene body. We could have that as an intron exon if I said intron equal true. The proximal intergenic regions, which are the ones closest to the genes, or the distal ones, which are the furthest away from genes. All of the ranges in terms of if you want upstream 5K or 5 kilobases or 2 kilobases, you can change all of that in there. But I find that just a quick and easy way to go, here are the basic regions of the genome. And also do it this way because if there's a transcription start site within a gene body, it gets called a transcription start site. So a lot of genes have got multiple transcripts with alternate transcription sites, and this calls them all as transcription start sites. So you will have a gene with multiple transcription start sites. I just thought that made sense. Okay, so we've got our genes, we've got a bunch of genomic regions organized, we've got our genomic information. With ChIP-Seq, we often have black or grey listed regions, which are parts of the genome we're not confident that what we're seeing is going to be reliable. The blacklists are available on GitHub somewhere. Just search encode blacklist and they will turn up. That's what I do every time. I've supplied it with the package. Grey lists are experiment specific. And when you've got an input sample, which is just generic IgG, then it gives you an idea for where things just look a bit weird in the genome and you can just mask those. So we're going to use these black and grey lists to mask bits of the genome that we don't trust. The function I've got there, import peaks, we'll just give it both of the black and grey list files. We'll allocate the seq info and we'll tell it it's going to be a bed file, and that will give you a nice GRanges list with both of those regions or both of those sets of ranges. You're all happy with that? So import peaks is a really simple function. You can give it multiple bed files, multiple narrow peak files, and it will give you a GRanges list. And one of the key little tricks that I've done here is when you give it the seq info object, only the chromosomes in the seq info object are parsed. Everything else gets ignored. So this is my way of throwing away anything on a scaffold or a haplotype or whatever in there. And that's why I kind of like setting up the seq info early. All right, so that was just so you can see what import peaks does. It gives you that nice GRanges list with any of the bed columns available. Are there any extra columns there? They're just ranges, I think, with bed files, I think. Yeah, cool. So what I would actually do to make these a set of ranges that we want to exclude is I would import them and then I'll unlist it, apply genomic ranges reduce, and that just gives us the reduced set of ranges that we want to ignore for our entire analysis.
That's something to do only for regions of the genome that you don't trust. Regions that you don't trust, or do you also include, for example, let's say you don't think transposable elements should be there, and you want to mask them as well?
Yeah,
or repeats in general?
yeah, no, that's an excellent point. I have assumed that they're in the blacklists. I haven't checked that. Often those regions are really hard to align to anyway. Um, yeah, but yeah, you could mask any transposable element you want. However, as long as you're comfortable, you've accurately defined a transposable element. They're fun little beasts. Yeah, so you can mask anything you want. Yeah. All right. So that's the setup. We've got our regions we want to mask. We've got our annotation sorted out. We've got our genome information sorted out. Um, we're ready to go. Let's do the fun bit. This is the analysis. I'm not wanting to show you how to call peaks with Max2 or any of the other tools that you may prefer. I know that there are quite a few alternatives. I used to use Max2, so it's what I've prepared these samples with, but please feel free to use your favorite tool. Each sample, we're going to have 6 samples that we'll look at in a minute, and they'll have their peaks called individually. And those peaks won't overlap perfectly. They'll kind of mostly overlap, or you'll see it in 2 out of the 3 or whatever. And so what we're going to do is we're going to load in those peaks and then just unify them so we're comfortable that they represent our best estimate of where the binding is for the entire data set. So as I've just said, the ranges of those peaks will just vary a little bit. Maybe it's by 1 base, maybe it's by 20 base, maybe it's missing. The center positions will be slightly different across samples. And we want to define these consistent ranges, often known as a consensus peak. Then downstream, we'll actually set them all to be the same width. There are no fixed rules about how to do this. I'll show you a possible way that I often used, but it doesn't mean it's the Bible, and it's the only way to do it. It's a vaguely sensible strategy, and there are other vaguely sensible strategies that exist. So let's just define our samples. And this is a big chunk. I'm sorry, it's a little bit confusing. I thought about trying to simplify this and make it really, really easy, but then I thought, nah, bugger it. Just do a really big one. And so there's a big chunk here. This covers our H3K27 AC samples and our ER samples as well. You can see them all there. So if you had just have a look at that samples object, do you guys want me to see this executed in front of you, or you're happy just looking at your own screens? I've just created a table and I've split it in 2. So one part contains the ER samples, the other part contains the estrogen receptor samples. It's all right. Ah, 3, and the input. Yes, thank you. I forgot about that. Excellent. Thank you. This is why I want people to, because the chances of me forgetting something just in that exact moment is high. So let's define where the peaks are. These are going to be prepared in narrow peak format, which is a Max2 kind of like standard. A lot of peak callers will output data in narrow peak format. It's very much like a bed file, but it has a few extra columns in it about how confident the caller was in that peak. So they're all named after the ID, just with an underscore peaks.narrowpeak.gz. And they are all in X data in the package. So we'll just import that very first peak, and you can or the first set of peaks from the first sample, and you can see what narrow peak format looks like just by looking at that. We've got the ranges, and we've got score, p-value, q-value, and then there is that column. Is it called peak or called peak? Yep, great. That's the relative position of the center within that range. So if you've got a range that's 200 base pairs wide and it says 80, then that's where it thinks the maximum of the signal is, and it calls that the center of the peak. Usually it's near the center of the range. All right. So what I do when I import things, I'll import all of them now, or we can import all of them now and call them peaks. But there's an argument there, or there's a couple of arguments there which are interesting. We've put the exclude ranges in the blacklist, and that means that any peaks that overlap that just don't get loaded. We've used the seek info to exclude any peaks on scaffolds or whatever. And I've put center equals true, and that adds an extra column called center, spelt the English way. Any Americans here? A long-running rivalry that the Americans are oblivious to. That will take the peak and it will change the center to being an absolute position, not relative to the start of the peak. So it actually turns it into a coordinate position. Then I'm adding the names. So we're happy with what that's done? Am I going too slow, too fast, talking too much? Okay, cool. So now we've got our peaks. We can have a look at them. Yay. The first thing we might want to do is look at the width of the peaks and just see how they track across these different samples. And I've put in a function plot GRL col, just plot a GRanges list and a column from the GRanges list. By default, it will plot the width. This width is a secret column that's hiding in your GRanges object. And so you can see across all our 6 samples, we've got 5,171, I think, is the sample with the fewest peaks. And then the third one along, we've got just over 11,000 peaks. So real discrepancy in how many peaks get called per sample, which is something worth noting. And that plot will tell you how many peaks there are by printing it over the median of the box plots. And then I think that's the 1th percentile at the bottom, but you can change that to being whatever you want. So 99% of the peaks are wider than 185 base pairs. It's a ggplot, so I've just changed the y-axis to be log10 because that works really nicely in this context. That function itself, you can do a lot with, and on the next screen, I've gone a little bit crazy just to have some fun because I like having some fun. I've just defined a vector of colors because we're going to be dealing with E2 and E2 plus DHT. So I've called E2 is going to be blue, and then with DHT it's red. And this one here, I'm just showing you can go nuts with this function if you really want. You can just use it to look at the widths, or we're going to look at the scores. I'm using a violin plot. And if we put our samples object as a data frame, we can use the columns as long as there's a column in there that matches the names of the GRanges list. And so there's a column in there called treat, and we can color them by treatment. And I've turned the total off by saying total geom equals none. So you can have as much fun as you want with that plotting function, depending on what information you're wanting to pull out of your peaks. Usually it's something like width and scores and p-values and those kinds of things.
Really big question. So on average, how many peaks do you expect?
How long's a piece of string? It depends on each particular chip target. Yeah, so in the H3K sample, I think they've got about 40,000, and they're much wider. Yeah, so yeah, it's just how many sites does the target bind to? If it's a really prolific binder, you'll get thousands. If it's a really niche targeted binding protein, yeah, it could be, yeah, could vary. All right, another little fun little uh, function, plot overlaps. This will pop out an upset plot, and this just shows you where these peaks overlap each other. And for those of you who witnessed the saga of ggplot changing to S7 last year, this was the function that I was having trouble with. It's no longer in complex upset, which is a lovely package. I love it, but it just became a little bit challenging. You're happy with that? Yeah, you can sort the sets just by ascending size or just none or whatever. This is kind of a fun little function because these peaks don't immediately overlap, exactly overlap, I should say. What actually happens under the hood here is it forms consensus peaks, like it just merges all the ranges and says, right, here's a range where I can see something. How many of the samples overlap that? So the peak numbers on the left-hand side won't actually match the true peak numbers because there's been a little bit of merging of peaks. So I wouldn't rely on those numbers there to be exact peak numbers because of that little merging process that I've had to happen. All right, so let's get into consensus peaks, which is where we want to look for confident binding sites that we think might change in response to treatment. We don't want binary on or off sites. We're not looking for sites where there was nothing going to something or something going to nothing. We're looking for a change. ChIP-Seq itself is very different to RNA-seq. A lot of people are familiar with RNA-seq, but in ChIP-Seq, assuming strict diploidy, which is not really an assumption in cancer cell lines, but you've got to get a maximum of 2 fragments at a site from each cell, unless there's duplications, of course. And so what you're looking at with changes is within the cell population, you're looking at how many cells have changed, not whether this site is more strongly bound in that one cell, because you can't tell that. It's about how many cells in the population are increasing binding or decreasing binding in response to treatment. We know a lot of cell lines, when we treat them, actually they're not completely homogeneous. They differentiate and they have different properties. So what we want to look at as sites that are confidently found in either condition. Maybe 2 out of 3 in either of the conditions might be a smart way to look at it. And once we've found those sites, then we might want to merge across conditions. And if you remember back, one of the samples had 5,000 peaks, another sample had 11,000 peaks. So we're clearly going to probably bias our consensus peaks to the samples that have more peaks in them. Limitation of the platform. All right. So the question is, how do we define the boundaries of these ranges, and what do we do with the peak sensors? And I've got some steps to solve those questions. So I've put in a function called make consensus, and here if we just subset out the E2 samples for ER, going to make the consensus peaks, saying that we want the proportion to be 2 out of 3 or 2/3 of the samples in that input. And by putting var equals center, we're going to retain that center column, which is why I went to the effort of making an absolute coordinate instead of a relative position, because once you've got different peaks, that relative position is going to not be helpful. And that comes out as a numeric list. And then down the bottom, you can see which sample it was in and how many samples it was found in.
You can ask a software design question. Is there a reason why you have to rely on the samples object so that you can map to the level EQ instead of just add this information to the ERP object? See what I mean? Because so you need 2 objects to work.
Yeah.
When you could have 1.
Yeah, I guess.
Could you have 1?
I guess I'd have to have in the metadata of the G ranges list, because that's probably the best way to do it. Sorry?
So you only work with 1 object in the end.
Yeah. Yeah. It's a good question, and maybe I could have extended an object class here to incorporate that, but I stuck with the existing object classes. Yes, I could have put it in metadata. That's a good question, and perhaps I should give that more careful thought. Yeah. Where am I? Okay. And the obvious thing to do after we've got those centers there is we might want to actually unify them into just being 1 coordinate instead of a list of 2 or 3 coordinates. And so we can use a mutate from ply ranges. And hopefully that syntax makes sense, that we can now just find the average position of those centers. So now we've got a consensus peak where we've averaged the center positions. There's no rule for that. You could do a median, you could do a mean. You could just choose, pretty hard to choose the one with the highest score at this point because we've lost the score, but you could keep the score column and mess around with it there. All right, so now we've got consensus peaks. We found a center position that is vaguely sensible, and we've got our E2 consensus peaks. So now this is me expanding this up. I hope this syntax is not too overwhelming. Um, I've taken the ER peaks and I've split them by the treatment group. So we're going to have two G range or a list of G ranges lists. One G ranges list will hold the E2 samples, the other will hold the E2 and DHT samples. And then within those, we'll make the consensus using 2 out of 3. We'll find those centers. So that's the L apply here. And then we're going to make it a G ranges list again. And then we're going to make the consensus again. So we've got the consensuses within the treatment group, and then we've got the consensus of the consensuses. There's no proportion there. It's just if it's in either, it's in. And then we're merging the center again.
I don't understand the line below L apply like this dash and then in parentheses peaks.
Yeah, that's the shorthand. It came in 4.2 or something, I think, but it's shorthand for the word function. Yeah. So those of us who are really lazy just do that now. So that's a big line. That's a big chunk of code, that one. There's a lot actually happening in there. So if you've got questions about it, then feel free. Now, this was me making consensus peaks in a treatment specific manner and then merging to make overall consensus peaks in a treatment agnostic manner. We could have just put them all together and just gone, all right, let's just find where it's in 4 of the 6, or which is 2 of 3 again. Or we could have done any other approach. That's the one I chose. Um, Often I would have kept those intermediaries and done other things with those intermediary treatment-specific consensus peaks.
So the resulting object has peaks that are in either treatment.
Yeah, that's right. Yeah, if we go back to the upset plot, I actually forgot to point that out. There's only one little intersection here. Oh, is my mouse there? Yep. You can see there's 128 peaks that were unique to the DHT treatment, and there were no consistent peaks that were unique to the E2 treatment. Yeah. So a lot of the peaks were, shared between treatment groups anyway. And I actually find that figure to be helpful in determining the strategy for you, what that you want to move forward. If you look at it and go, oh, look, they're all pretty much the same. Let's just go two out of every 3 in either treatment group. That'll be fine. You could do that. Okay. All right. So that's given us our final object here, which has a consensus peak where the center of that peak is estimated to be by doing all those means of the centers, and then which of the treatment groups it was a consensus peak in. I think there's an end column at the end there. Now, one final step, I've added one line to that entire chunk just to overwhelm you even more. And I'm now going to add a region column. And this is saying, what's the best overlap? With those regions we defined, with the promoters, the upstream promoters, each of those peaks, what's the best region that matches that? And so that will now be in a column in the data set. I did that separately to show you the first process and then the mapping to the region. So now we've got peaks and we know whether they're in a promoter. We know whether they're in a gene body. We know whether they're miles away from a gene. We could put any other regions we want as well. We could have got some enhancers from ENCODE or something and put them there, and whatever features you want to do, you can just put them in.
When looking for overlap, do you have like a priority list, or do you look at centers, or how do you determine?
It's the maximum proportion.
Okay.
So prop overlap is the underlying function. So if you've got an overlap of 51% and 49%, I don't know what I'm doing with ties. Can't remember. Pretty rare occurrence. But yeah, I have to go back and check my code. I would have thought of it at the time. Okay, so that's our object. And we've got our region there sitting on the end saying that that's what each of these regions are ranges or peaks best maps to. And now I've added one extra line. I forgot. I forget how what I've written sometimes. And this time we're going to map those features to a gene. So we're not only going to know what kind of region it maps to, what's the best target for it. And so I've put in the function there, map by feature, where we give it our genes from our split GTF, we give it the promoters from our regions. If we had high C interactions, you can put them in as well. If there's enhancers, you can put them in as well. We don't have them today, but we can just pop them in, and that will choose the best feature or the best gene, sorry, that will map. And it's called map by feature because if you've got an enhancer, it will map differently to genes that are close by. If there's high C interaction or interactions, it will map differently because if you've got direct connection, it will use that. There's a help page there that describes my mapping process. So now we've got our peaks. We've mapped them to what type of region they're bound to, and then we've got all the genes. Some of them will map to multiple genes because we know there's often genes on overlapping strands or the other strands, and what gene they map to. They're all as character lists, or I think they're character lists. So.
Maybe I missed that, but could you explain again how you define promoter?
In that function define regions. Yeah, I've gone, the default is 500 downstream, 2500 upstream, but you can change that to any value you want. And then I've set upstream promoter is up to 5 kb upstream. And so they're the default ranges.
For example, where the TSS is, so that when you define like an interval downstream that you don't go over, or if you say like 500, it's always going to be 500 no matter what's there.
Well, it's your preference. I don't think anyone agrees on how big a promoter is. If we went around this room, we'd all have a different answer. So you can set that however you want. Yeah, because promoters can be downstream from the TSS.
Sounds like you say 200, and most of the genes, it's fine, but for you 10% or 5%, it's going to be over.
Yeah, I know. Yeah, there's no perfect solution. Yeah, I think if someone comes up, like, I mean, you could actually define those promoters by going to the Phantom 5 project or something along those lines and try and get better promoter representations. Yeah, absolutely. Yes. But this is okay, all we've got is ChIP-Seq, we've got a GTF. What's our best strategy? But yeah, you can source all of that information if you'd like it. All right, here's what our consensus peaks look like. If we run it into plot overlaps again, this time there's only 2, so it makes a Venn diagram for us. If there's 3 or more, you can make an upset plot. If there's only 2, it makes a Venn diagram. So are we happy with what I've done there? I've just quickly made a G-ranges list. One is the E2 consensus peaks. The other one's the E2 DHT consensus peaks. And I've used those columns, E2 or E2 DHT, to filter. So you can see those consensus peaks generally consistent. There's a few where there's some less confidence in either of the treatment groups. I put in another little function there because I like to antagonize Hadley Wickham, who doesn't even know I exist. But go the pie charts. He hates them. Um, yeah, so we can now just look at the consensus peaks and where they bind by region. I think this is quite informative. So there's my little function there, plot pi. And those labels are incredibly manipulatable. I don't know that exact English word, even though it's my native tongue. But using glue syntax, you can absolutely mess with those labels as much as you want. We'll do that when I do the donut plots. I like pies and donuts, so I've got a donut plot coming as well. All right, so that's the peaks. We've had a quick look at the peaks. We've looked at where they've bound. We've mapped them to genes. We've kind of done all the foundational stuff. Now we're getting into the differential signal analysis. I'm just trying to make sure I don't go too far over time. So this is where we're seeing if these targets are coming on or off of sites across the population in our samples. So where does it change? This is always an incredibly dynamic process. I think these samples were done at the 4 hour time point because they assumed that any changes you saw at 4 hours were going to continue and continue moving in that direction. Who knows? There are transcription factor dynamics which are incredibly different. The androgen receptor sits on the DNA and just sits there. The estrogen receptor comes on and off in a cycle. So there's all sorts of different transcription factor dynamics going on. There's a lot of complexity to this. We're just going to ignore that and just say, where is it changing en masse? And that will help us identify what genes are likely to be impacted. So I call it differential signal analysis as opposed to differential binding analysis. Has anyone here used the package diffbind, by the way? You come across it? Yeah, I've played with it a fair bit. The reason I did I explain why I wrote this as opposed to just using diffbind? Diffbind didn't play, yeah, I did. Didn't play well with pipe and it used data frame objects instead of GRanges list. So the stuff I can run on my laptop with extraChIPs, I can't run on my laptop with diffbind. So when we do differential signal analysis, generally the strategy is we set fixed width peaks and we set all of the peaks to be the same width, and then we just look across samples. And also, if we set them to be a fixed width, it does help us as we do sequence analysis because a lot of sequence analysis tools want fixed width across all of your sequences as well. So setting fixed width peaks is really quite helpful for downstream analysis. So now we've got our consensus peaks. Let's set them to the center and make them 400 base pairs wide. There's a couple of little tricks I'm doing here. So we've taken our consensus peaks. We're going to use mutate and create a new column called range. And we're going to put the chromosome names in there and then the center position separated by a colon. So that's going to have chromosome 1, colon, whatever that center position is. Then I've written another fancy function called Coulter ranges, and we're going to move that range to the genomic ranges component. So we'll end up with a single width range, which we then resize to 400 kilobases. I could have perhaps wrapped that into a single function, but I like the transparency of that. Coulter ranges works really nicely with a data frame. So if you've got a column that has a GRanges in it, you can just call Coulter ranges and we'll give you a GRanges object with that as the range. Yeah. Now, when we're doing our differential signal analysis, what we're all actually about is count. So we've got how many alignments within that peak and do the number of alignments count. So there's a lot of similarity to RNA-seq at this point in time. The BAM files were too big for the workshop, obviously. So I've pre-prepared those and the code is in the workshop here, showing you what I did. I think I've got a big do not run next to it or something. There it is. That was the code. I ran it on my laptop. So 16 gig. It worked fine. Yeah. Okay, so we can just bring those peaks in those counts in using the file I've provided. It's just ER_SE because it's a summarized experiment with the counts from the estrogen receptor. And then there's two little commands there just so you can have a look at it. And you'll see that those row ranges are exactly the same genomic ranges object you had that you were working on. The cold data will have the totals. Am I going to talk about that in a minute? Yeah, I am. Good. Now you might, yeah. So the totals that you see in the cold data, that's the total alignments in the BAM file, not the total alignments within peaks. And that makes a really important difference when we come to normalization. A use case, well, analysis I used to do a fair bit in this group was looking at the androgen receptor. And the androgen receptor is sequestered in the cytoplasm until androgens are present. And so you can have it in the control treatment group along with your E2 and H3K because they're all coming from the same treatment, but there will be no binding. And then you hit it with androgens, E2 changes, H3K27AC changes, and AR goes from nothing to 100. So it's very different to like an RNA-seq type context where you can have a treatment group with absolutely no binding, or next to zero binding, and a treatment group which is a bunch of binding. And so it's a very different kind of data structure. We'll talk about that a little bit more in the normalization steps coming. And that makes what you choose for your library size makes a really, really big difference. If you choose the peaks to normalize to where there's no binding, the 5 peaks where you've got binding will look like they are the strongest peaks in the history of the universe. And so it's really important to use the complete library sizes, not what you find within the peaks, unless you're really confident that they're the same distributions. Generally, people don't. Okay. You can quickly just have a look at this. This is a really hack version of the fraction of reads in peaks. You can formally actually calculate this using, I think, ChIP-QC. There's a lot of other packages that do this. This is a quick hack just to show you that within all these samples, we're down to about 2% of the counts actually come from peaks compared to the total library size, and that's not unusual. Different chip targets will have different profiles. The noisier data sets will have different profiles. Generally, the guide says FRIP above 1%, and you're pretty good. There's an ENCODE guideline about that. I think I've linked that in the main page. Just to kind of show you that the totals we get there and the totals in the peaks are very different numbers. So counted is actually the peak summary. Yeah, so the first one we've got 16 million alignments and 285,000 of them are within peaks. So very different to RNA-seq. So this is a summarized experiment, and we can add any assays we want because we love working with summarized experiments. So I'm going to add a log CPM assay. Now there was a glitch in the row names, so I just had to set the row names back manually. They came up as blank, I think, when you do the CPM, and the summarized experiment didn't like that. So, because summarized experiments are really good at checking that everything is where it should be, and 99.9% of the time that is excellent. Every now and then frustrating. So here's a couple of functions. I haven't got the figures up. I'll leave them for you to create. So we can have a look at the assay densities. So plot assay densities will work with any summarized experiment object. So we can just have a look at the counts. We can transform it using the log1p transformation. Because we now have the metadata correctly in the object, we can use one of the cold data columns treat to color these. So we're coloring these densities by E2 or E2DHT, and then just adding our ggplot colors. And then when we're creating log CPM values, what we're actually doing is we're normalizing it to library size. And that's a kind of an assumption we can make there. So we've got our raw counts, and then we've got our counts normalized to library size. So handy little functions. Again, I use those ones all the time. If I'm working on different data types completely and I've got a summarized experiment, I go back and use these ones pretty regularly. I know there are plenty of other packages that have got assay plotting functions from summarized experiments. I like mine. And then we can check the relative log expression. Relative log expression is a great little QC step. So the first plot here, we're just going to look at this on the log CPM values. What it does is it basically creates the median, subtracts that, and then looks at the distribution around the median. everything looks great here. So there's nothing to see, but it's a good check. But there's a little trick which I find really handy here, is that we can do relative log expression, grouping it by treatment group. So I've put by X equals treat, and that will just see if there's any global differences between the treatment groups. Just a different perspective on the data. It's you know, coming up with as many different ways to look at the data and find any possible problem we can is a good strategy in my brain. And then we can just plot a PCA of any of the assays we want. Obviously, the log CP-M assay makes a really nice one for plotting PCA, and it'll automatically come up with labels. You can turn them off. You can manipulate these as much as you want, but again, a nice little PCA plot.
Does it take all features or only the top X%?
By default, it will plot all points, but you can set it to and we'll do that later, but it's random sampling rather than top features. And perhaps I should add that as a capacity. Yeah. Yeah. When we get to the HDK data and we've got genomic sliding windows, that takes ages to plot. And so you just downsample that. But yeah, I could actually set that to be the top features. That would be smart. Thank you. Okay, so we've looked at our data, we've looked at the distribution of the counts, we've looked at the RLE, we've done the PCA, we just go, all right, data looks great. I'm really looking forward to doing this analysis and getting some results. Yay. So before we get into our actual statistics and the analysis, we want to think about normalizing our data. I've already mentioned the counts within peaks. So we generally, if we don't normalize on counts, we'll just use library size. That will automatically happen under any of the negative binomial models. It just uses library sizes as counts. That's how it works. But we can also have the scaling factors from TRMM or RLE or any of the other ones that are available in EdgeR or DESeq2. I've used the EdgeR versions here. So, upper quartile is another one. And then there's some newer ones which have been implemented I haven't used because they're more single cell specific. But we can use those, but we want to make sure, and this is key with ChIP-Seq because it's very different to RNA-seq, that we've got the same distributions across treatment groups. That's really quite important. And the assumption of TMM and RLE is that these are roughly the same. And I've given that example. So to check what we can do for normalization, there's a great little package called Quantro. Stephanie Hicks and Rafael Irizarry put it out about 10 years ago, and it tests for equality of medians within these distributions. You can do permutation tests using it, but with 6 samples, that's a meaningless permutation test. But once you get up to 8, 10, 12 samples, those permutation tests really can be really quite powerful. So we can just grab our assay. It takes a matrix. So we grab our log CP-M assay. We're grouping it by the treatment group, and we're testing for equality of medians within those treatment groups. The ANOVA p-value, I never know whether it's ANOVA or ANOVA. I say ANOVA because that's just my accent. 0.9. So obviously we're gonna keep H0 that the medians are all equivalent, and that these are drawn from the same distribution. So that means we can normalize it however we want. And when we fit the data, we're going to use a function called fit_assay_diff. And the methods that have been implemented here are the GLM quasi-likelihood fits from EdgeR, the negative binomial Wald test from DESeq2, or limit trend, which just takes a little bit more care. We're going to do that later. But yeah, the other models we can fit. I haven't put in Voom. I just felt more comfortable with limit trend. I had a good reason at the time, but I wrote that function probably 4 years ago, and I can't remember what my brain was thinking at the time. Limit trend is fitted on log CP-M values, whereas the others are fitted on counts. The diff-bind standard is RLE normalization and DESeq2. So if you've used diff-bind, that's pretty much what it uses for the defaults. I've gone through and checked my results very carefully against those just to make sure I haven't done anything stupid. Non-zero probability. I've known myself for a long time. And my results are identical. to diff bind. So I'm really confident that I've implemented the models the exact same way. So if you're familiar with diff bind and just want a more computationally method with lower overhead, you get the same results using extraChIPs and diff bind with the same normalization settings. I've also implemented the treat methods. Are people familiar with the treat methods, or am I sounding like I'm selling you lollies at that point? Okay, there's people confused. Limma and EdgeR have a method called treat, where instead of testing for is there zero change or non-zero change, it's test the range. So is the change within this range? That's a rhyme. A good thing I can't do hip hop. So is the change within that range, or is it outside of that range? So it's a range-based null hypothesis. I find them incredibly useful, and I would do that by default, but I haven't done it here. So that's implemented as well for any of these tests. For LMFit, it calls treat. For GLMQLF, it calls GLMQLF treat. And for DSEQ2, it calls the equivalent function in there as well. So we fit. I'm Australian, so I grew up eating and drinking limma. So I tend to work in a limma-centric method. So we'll design a model matrix here, which is very similar to how they are used in limma. And so here we're going to just take our cold data and we're going to respond to the treatment. And then we're going to call fit assay diff using that design matrix, telling it what normalization method to choose. I've chosen TMM, could have been RLE. Do I want the results as a summarized experiment or as G ranges? I wanted it as G ranges, and I'm going to fit using the quasi-likelihood methods of EdgeR. So fit assay diff will do that from the summarized experiment, and then add diff status just adds a column, which tells us whether it's increased or decreased or unchanged. And you can set that to be up, down, or whatever words you'd like to choose. So that's how we would fit models in the extraChIPs approach. And yes, if you are familiar with summarized experiments and you just like to fit standard DSEQ data sets, you've got a summarized experiment, so you can fit it using those methods. If you like, just being careful with your normalization strategies that you're not accidentally choosing the peaks. That's why I like to have it slightly set aside from that, so you don't fall into a very easy trap to fall into.
I have a question just to hear your opinion about that case, which is beyond the scope of this example. What do you do for normalization in the case of global shifts in data, like going from a case where you have 1,000 binding sites
Great question.
to 20,000 binding sites?
I love that question. I'm going to look at that in a minute.
Okay.
This data set is a very well-behaved data set, so it's just an example of what you can do. The default would be just to use library sizes and not have any of these scaling factors, and that's the best you can kind of do in that setting. What I like to do, particularly in a sliding window context, is Q-smooth normalization. Anyone here come across smooth quantile normalization? Another one of Stephanie Hicks's packages. Sorry, Dan over here. Yeah, that does quantile normalization, but in a treatment group specific manner. And so you can normalize across groups, and there's a weighting method that they've developed where you can normalize this region of signal as being from the same distribution across samples, but then you can separate that signal out and normalize within treatment groups. It's pretty slick. Yeah, smooth quantile normalization. That's so I like that with sliding windows. I remember microarrays. Anyone else here remember microarrays? 3' microarrays? Yeah. I did my PhD on exon arrays. By the time I finished, you couldn't even buy an exon array. It took 10 years. What was I going to say? Yeah, so quantile normalization was a big thing in microarrays, and it was really helpful, I think, in that context where you have, I think it was 11 probes to a probe set, and you quantile normalize your probes and then merge to a probe set, so you didn't get any over normalization artifacts because it kind of averaged itself out a little bit in that probe set merging thing. And so sliding windows are analogous to me in ChIP-Seq. That's just my opinion. All right, so we've done our differential signal analysis. That was a massive side tangent there. I'm sorry about that. Someone asked me a question that got me excited. I'm so excitable. All right, so we're going to just define some colors. I like defining colors, vectors of colors at some point in time so that every plot I make has the same colors for the same thing. Often it's for treatment groups. So I defined region colors earlier because every time I do a plot with regions, I want the promoter yellow, I want the gene body green, very similar to Chrome HMM tracks. Oh, you could use Chrome HMM tracks instead of the defined regions, whatever you want. I originally wrote it thinking about Chrome HMM tracks, but then I didn't have it for a lot of the cell lines I was working on. So stay focused, brain. Okay, so here we are. We've got our differential signal results. Converted it to a tibble using my little parser function there, stuck it in a ggplot, log CPM, log FC, classic Ebane plot. We're all pretty happy with that. Looks good. There was no weird artifacts and bias. When you look at AR in this kind of thing, it's really different because it goes from nothing to a lot. And so you get really different shapes. It's quite interesting. Don't have any here. So you can just grab MA plots. Oh, there it is. Looks good to me. What have I done? I've done low S. Yeah, I'm fine with low S. I often get those little kicks at the low end. They never bother me. It just means there's a few low signal points that are upregulated. It's the overall bias that I'm interested in. Volcano plots, we can do the same thing. Oh, I've got static colors again. You can ignore that line. I copied and pasted. So a volcano plot. It all looks pretty similar to how you'd expect it to look. And then you can do my favorite donut plot. Huge chunk there. Plot split donut works really well. As long as you tell it what you want in the inner region and the outer region, it'll give you a fine plot like that. But this is me customizing the crap out of it. And you can see the glue syntax I've got there. You can call the data inside the function as .data for the inner or the outer rings. You've got N and P as the number and the percent, and you can format those however you want there. So reasonably, cumbersome, but you get great looking labels. Have they come out in white for you guys? I found a bug about a month ago and it's pushed to Devel, but if you're doing this on release, those labels might come out black. So I'm sorry about that if you're seeing that. But there's the donut plots. I find them really helpful just to get a quick little picture of what's changed in what regions. Okay. We're not going to get through much of this because we've got 15 minutes left. But this is a completely different approach. And I wanted to do as much of this as I could get to. So H3K27AC is really, really different to a chip target like the estrogen receptor. The estrogen receptor is going to bind to a point on the DNA, whereas histone marks can cover huge ranges of DNA. So this might be a little bit more similar to a TAC-seq or some other different platforms. I think P300 I've had a look at was pretty similar. So you get these really different peaks. And there are different types of peaks. Generally, if you've got a fixed width peak for the estrogen receptor, if you see change anywhere in that peak, it's going to be relatively consistent within the peak. It's bound here, it's changing, it's going more or less across the tissues, the cells within your sample. H3K27AC is really different. It's not just a smooth line or a peak. It's got valleys where there are other transcription factors bound, and just, the ratio of signal is a really different shape. And that means we want to think differently about how we do differential signal analysis. When we had a peak, we could kind of go, here's the center, let's go 400 base pairs. Any change in there, we're going to pick it up. When we're dealing with like a 10 kb a region for H3K27AC, that's a bit of a different question because it might be the tail end of it that changes, or it might be in the middle that changes. And so finding the region, I think sliding windows are much, much more suitable to this kind of peak. We'll define sliding windows across the genome, and then you can tell that exact region that changes without having to define any peaks. So this is a peak agnostic approach. You don't need max2. Having said that, I'm going to use a lot of peaks, but you strictly don't need them. So the sliding windows was Aaron Lunn and Gordon Smythe did a huge amount of the background work to making this work, and I've just wrapped a lot of their functions to keep it consistent. And I've introduced a new method for merging ranges, the sliding windows. I'm merging by the harmonic mean p-value instead of Syme's method, which Gordon uses, or you can choose maximum signal or other methods. So my default method is using the harmonic mean p to merge across overlapping windows. Okay, so here's the code to import the peaks. These peaks were created by Max2 call peak, merging all 3 samples from a treatment group. Max2 can do that. So instead of going, here's an individual sample, call the peaks, you can also go, here are the 3 samples from the E2 treatment, call me the peaks. And that will actually weight by coverage, so you don't get that bias that we saw with the 5 to 11,000. Again, I've chucked some glue syntax in there for the names. That was the typo I found. So it might look a bit different on your workshop. If it doesn't pull off the .narrowpeak.gz, then sorry about that. It meant to, but then I changed the file names and I didn't change the code. Okay, so here's what the peaks look like, and these are really different to ER. You can see we've got nearly 50,000 peaks here instead of the 5 to 11,000 that we saw, and they're much bigger. Instead of hovering around the 300, a median of about 300 base pairs, the median around here is about 1,000. So it's 1 kb pushing up to 30 kb. Really, really different kind of a signal here. So we're going to use sliding windows, relying on the heavy lifting of Aaron and Gordon's work, and using ChIP-Seq, the code's there for you to look at. It throws out any windows with low count, so you don't have the entire genome, but you've got most of it. I was able to do this with 6 BAM files on my little 16 gig laptop. If there were 7 BAM files, I think it would have struggled. So it can run, and that was just purely using the ChIP-Seq infrastructure. When you've got the genomic windows, ChIP-Seq's got a lot of options for filtering out those windows to throw away the sliding windows that are uninformative. There's just not signal. There's something there, but it's not really relevant. This is similar to throwing away your undetected genes in RNA-seq. The approach that I've brought in here is dual filter, where you can say, here is a set of ranges where I expect to see signal. Use those as a guide, and then set some thresholds in terms of the ratio above input and the minimum number of or the minimum coverage. So taking these reference ranges, now those ranges are those peaks that we've just loaded. We expect to see signal there, but you can pull them in from any data set. A colleague was looking at RNA polymerase 2. She's going, how can I use dual filter on RNA pol 2? How can I define ranges where I'd expect to see that? So we're trying detected genes in that cell type, because that's where we'd expect it to be, or maybe promoters. So you can use any set of ranges that are appropriate. You can pull them down from ENCODE or from any other place you'd like to. And you set a value saying retain 50% of the peaks, which is a really restrictive setting, or 90% of the peaks, which is, a really inclusive kind of setting. I usually find around 0.7, 0.8 works. This was also really resource intensive. I did some nice some fun debugging on this about a month ago, and I couldn't run it on my laptop before I did the debugging. I accidentally introduced a copy on modification massive. When you copy on modification on genomic windows, it blows your memory out, but I sidestepped that, which was quite wise. And so now I can do the dual filter on my little 16 gig laptop, so I was pretty happy with that. I had to massively bump up swap though. So it was my little machine was at its limits. But I'm trying to just show you this saying this actually can run on a little laptop. So you can do like quite large data sets with extra chips using Seesaw and other functions on a little laptop now, which is good. Has anyone got like is 16 about what you guys have got, or you guys are all running 3264 because you've got money? Yeah, yeah, there's the nod of guilt. Thank you. Yeah, yeah. But yeah, you guys are coming. All right. So we can load in the counts. We've got about 5 minutes left, so this is going to be really fast. Um, load in the counts, add a log CPM assay in there. Same old thing. Checking our densities, checking the RLE, checking the PCA, and then we'll do the Quantro test again to check out what normalization strategies we've got. Now, importantly, we've got genomic windows here. We've thrown away the ones where it's not signal, but we're going to have overlapping windows. So again, a bit of a different dynamic to a discrete peak and a discrete peak. But just always good to understand exactly what we're looking at. Again, we're pretty happy these are drawn from the same distribution, but I'm going to demonstrate the smooth quantile normalization here because I think it's really helpful. I would use this for the androgen receptor if I was using sliding windows with the androgen receptor, but that's more of a peak type setting as well. So here's how we do smooth quantile normalization. I've created an object there called a QS, and I've just done QSmooth on the assay, and I've set the groups. And so that's what it uses to create the normalization weights. There's a comprehensive paper on that, which I'd encourage you to read if you're interested in these types of normalization approaches. And then I've added it as an assay called QSmooth, just by grabbing the QSmooth data from that object. On the workshop, I have a figure showing you the weights. All of these weights up around 1. So this is the output from QSmooth plot weights, which is telling you that it thinks that the distributions are very, very similar across the entire range of signal. When I do something like the androgen receptor, I see a very different shape, and that's telling you that you've got really different distributions. I think this approach is quite robust to different distributions. So I think it's a really nice approach. Standard thing. And this time we're going to do differential signal analysis using limit trend. Because we've got normalized log CPM values, we're in pretty good shape to use limit trend, but we will need to set the normalization to none because we've already normalized. We're going to merge the overlapping windows using the harmonic mean from David Wilson's paper. And I'm using a weighted approach to give you estimates of log fold change and log CPM. And I'm using the inverse of the p-values from each window to weight the estimates of log fold change. So you're going to bias it towards where you're seeing the most significant change. You can turn that off, but I just thought that made sense. So here's how I would do the limit trend without normalization. Hopefully that makes sense compared to what you saw earlier. So we're fitting the assay differences using the assay QSmooth, no normalization, and limit trend as the model fitting. Going to do the same type of approach. First up, we're going to merge by the harmonic mean p-value. I've got some wrappers to to Seesaw in there just to keep the output standardized in format. The Seesaw stuff is really good, you know. Aaron and Gordon are two of the, intellectual genius giants of the bioconductor community. So they're really good. But I just did it for consistency. And then we've just added the overlap, the region and the genes, mapping it to the merged windows again. We can do the standard stuff, the MA plots, volcano plots, and I went a little bit silly. We're running out of time, so I'm going to rush now. Here's the MA plots broken by region. So you can see that tend to get a lot of the change in the gene bodies. which is interesting. Probably a higher signal in the gene bodies as well. Then you can have a look and see using the volcano plots, break it down, see which promoters are having change, because that's what people usually care about, promoters, or upstream promoters. So by having all that information there, it's kind of nice. Again, we can check for where the changes are. Most of the changes are happening in gene bodies here using the split donut. The bit that I'm going to have to rush over is the integration between targets. And this is one of the things I was aiming for as I wrote the package. So here we can plot the log fold change for both targets for overlapping windows. And you can see in this figure here, where they're both detected, you can see that they're changing very, very similar, which is what we'd expect. But the box plots above have a little bit extra and to the side have a bit extra information, because you can see that top light gray panel, that's what's happening to ER where H3K was undetected. So you can't show those points because there's no H3K value, if that makes sense. And then over on the right, you can see what's happening to H3K27AC where ER was undetected. And there's nothing to see there, but sometimes there is. So you get a perspective on where they are, and that was using the function plot pairwise. Sorry about that. Where you can just take a GRanges list of length 2, tell it what column to plot, and it will automatically figure out which windows, or which ranges overlap, and do the best estimate. There's another little function I've included here called map GRL cols, where you can take a GRanges list of any length and give it a column that you want, and it will pull out those values and then map them by the name of the GRanges list. So you can see here, I've mapped the status, the region, and the log FC columns, and the ER prefix has been added before them. So you can map the status, map the regions, map the log fold change, and then the gene name is going to be consistent. I'll put that in collapse instead of var. Handy little function, and then split donuts showing where they're both changing together or not. The final thing in the closing second is I've got a function called plot HFGC. I'd really love you to have a look at that and have a play with it. The idea is to plot high C features, genes, and coverage. And there's the full plot at the bottom there, designed for exactly the results of this kind of analysis. You can have as many chip targets as you want there. But at the bottom, we've got ER annotated with a little red bar saying that's where it was statistically found to be increased. And then the track below that is H3K27AC, and you can see the windows I've highlighted in the middle where we've got change in both, what the genes are and what where they map to. So these are mapping to a promoter by the looks. So handy little function. If you've got results and you just want to go, look at this peak, look at this peak, look at this peak, look at this peak, just set yourself up the objects and just scan through, 20 peaks in no time at all. All right, we're out of time. So I finished there. Thank you, Maria.
I'll just say I've made a thread in Zulu for the extraChIPs workshop and put the material in there. And if anyone has questions, after this, or
please hit me up. Yeah.
In there. Okay.
So yeah.
Place for you to go. And yeah, just remains to thank Stevie for an excellent job.
Thank you.
Thank you.
Yes. I've got stickers here if you want them as well. I brought someone from one of my other packages, NGS reports as well, if you want them, and some BIC Asia stickers.